Now we have Javiera Ortiz Severín from INTA Universidad de Chile with the talk exploring antibiotic resistance in marine bacteria using phenotypic and metagenomic approaches. So please, Javiera, the stage is yours now.
Just give me a minute to is it yeah, so do you see it, the complete presentation?
We see it, yes.
Okay, thank you. So first of all, I would like to thank the organizers for inviting me to share my work in this symposium. I'm really honored to participate here with such excellent work has been shared by amazing researchers. And I'll add that I'm recently working on the Institute of Nutrition and Food Technology at the University of Chile. So how does studying the marine bacterial communities relate to this? This is through the One Health perspective that recognizes the connection between animal health, human health, and the environment, and aims to sustainably balance and optimize the health of ecosystems. The health of ecosystems through mobilizing sectors and disciplines in different areas of the society. So the goal is to produce safe and nutritious food, contributing to sustainable development. And this is where the research is important, both basic and applied research, considering that food is fundamental to life, but it's also a business. And one important source of food is fish, that is considered a good source of proteins and healthy fats. But overfishing worldwide has made unsustainable the catch of wild fish. And this led to the potentiation of aquaculture, where farmed fish production has increased over time, with China, Norway, and Chile leading the production of marine farmed fish. So Norway is the first producer of farmed salmons, and Chile is the second one. However, the history of salmon farming have been different in both countries. It is now generally accepted that most ecosystems may contribute to the selection and spread of antibiotic resistance, and our food and related environments could be a source of the transmission of antibiotic resistance. So the One Health perspective focuses on the risk assessment of emergence, transmission, and also the maintenance of antibiotic resistance and the interface between humans, animals, and the environment. So for producing 1,000 tons of, this is official information from the Pesca in Chile and the National Health Service in Norway, and states that for producing 1,000 tons of salmon in Norway, they use less than 100 kilograms of antibiotics, while in Chile, we use consistently over 200 tons of antibiotics. And this extensive use of antibiotics occurred due to high prevalence of a bacterial infection that caused high mortality in the Chilean salmon farms, but not in Norway. This infection is the salmon rickettsial septicemia caused by Spiza rickettsia salmonis, which is found in different countries in the world, but also only causes high mortality rates in Chile. So how come this happen? And here is where the study of the environment could give a clue about what is happening with the salmons reared in Chilean coastal waters. So by evaluating marine environment, who lives in association with the salmons, we can begin to understand the underlying process leading to the fish disease. Most previous studies on the impact of aquaculture in the environment have focused on sediments and associated bacteria after their isolation. The surface seawater environment has been less studied so far. And despite the important perturbations on the marine microbial communities as a result of the anthropogenic disturbances, this is an important focus. So in this project, we compare the marine bacterial communities associated with fish farms which are in direct contact with the fish, and in an area that has been historically saturated with aquaculture, and compare it with communities from clean or controlled sites, or an unpolluted site at a genomic and phenotypic, with a genomic and a phenotypic approach, using taxonomy and phenotypic assays to compare both sites, with the aim of assessing the antibiotic resistance risk of the bacterial communities in both environments. So in Chile, the salmon farming is circumscribed to the southernmost region of Chile. This is a map of Chile, and this encloses the Los Lagos, Aysén, and Magallanes regions, the most southern regions in Chile. And here are the regions, Los Lagos and Aysén, with most active concessions. So every red dot, it's a concession for a salmon farm. And finally, this is a map showing the region where salmon farming began in Chile, the Los Lagos region, over 30 years ago. And here the salmon presence is intensive and chronic. So the yellow points show where the samples were taken in this project in an area that has been surrounded by farm sites, and another one far away and near the Humboldt Current that comes from the Antarctica. Here, the aquaculture farm site that I just mentioned was considered under high anthropogenic impact, mainly from the presence of more than 300 aquaculture concessions in the area where seawater samples were collected. On the other hand, the Pacific Shore site was over 45 kilometers distant from the nearest fish farm and with no aquaculture activities. However, certain level of pollution could be expected due to the presence of ships, tourist activities, and river pollution. So the marine bacterial communities in this Pacific Shore site and the aquaculture farm site showed similar trends with regard to the most abundant phyla, like Proteobacteria, Bacteroidetes, and Actinobacteria, and Cyanobacteria. And the orders SAR11 and SAR86, and Rhodobacterales and Flavobacterales, which is consistent with the fact that they are the major constituents of several marine systems. However, principal component analysis show a marked separation between both sites. Although bacterial diversity was similar between the sites, the observed richness was significantly higher in the Pacific Shore site. In the aquaculture farm site, there were enriched amply consequent variants, or ASVs, that belong to order Pyrellulares, Thiomicrospirales, and Marinimicrobia, whose members are known to inhabit low oxygen habitats and facultative hydrocarbon degraders of orders Parvivaculares, Cordimonadales, and Oceanospirales. This suggests the development of epoxic areas, which correlates with eutrophication caused by nutrient enrichment from that could be due to intensified aquaculture, and the presence of organic pollutants, such as petroleum hydrocarbons, which are known to accumulate in the surface microlayer. And the vast majority of marine organisms are described as not culturable or had not been yet successfully cultured in laboratory conditions. And cultivation independent approaches have revealed that alpha Proteobacteria represent one of the most abundant marine bacterial classes, while most cultured organisms belong to gamma Proteobacteria. Here we observe something similar in the Pacific Shore site, but interestingly, in the aquaculture farm site, there was a high proportion of alpha Proteobacteria that could be cultured under our conditions. But not only this, but an overall increased diversity and richness was also observed in the culturable bacterial community from the aquaculture farm site. So in this work, bacteria were grown as a consortium in bacterial communities rather than isolates and subject to antibiotic treatments afterwards. with antibiotics used by the Chilean salmon industry. We were able to observe the presence of bacterial taxa that normally do not grow under standard culture conditions, such as cyanobacteria, marinomicrobia, clade 1A, NSA4, and NSA5 marine groups, which were able to grow as a consortium with the marine cultivar community, even in culture media that is routinely used in laboratory conditions. And these results suggest that our culture approach has increased the diversity of the culturable bacteria. It has been previously shown that co-cultures of environmental bacteria allow the culture of fastidious or previously difficult to grow bacteria, since the co-cultures can provide growth-promoting factors, enabling bacterial growth under these unfavorable conditions. So in the salmon farm site, the frequency of resistant colonies was significantly higher in the aquaculture farm site community, irrespective of the media used and the antibiotics supplemented. Regarding the taxonomic composition of the resistant communities, differences were detected among the sites, media, and antibiotics, but by integrated cultural enrichment and 16S RNA amplicon sequencing, we showed that the composition of the culturable fractions differed greatly from the environmental samples. So the antibiotic treatment was the main driver for changes in bacterial composition. Bacteria was grown both in nutrient and saline media were grouped together, but they separated from the antibiotic treatments. Also, the antibiotic treatments allowed the growth of a more diverse community, showed here, in the aquaculture farm site only, as reflected by an increase in diversity and richness. Those culturing bacteria did not erase the differences observed between the two environmental samples. And when predicting the antibiotic-resistant genes in the metagenomes from both sites, a higher amount of genes were found in the Pacific Shore site. Also, together with the gene counts, I know that you can see it here, but this shows similar trends in both sites, both in gene counts and in abundance between the Pacific Shore and the aquaculture farm sites. But when we grouped the individual antibiotic-resistant genes in antibiotic families, we observed that the accumulative normalized gene abundance was higher for the salmon farm sites, so the green ones here, for most of the antibiotic-resistant families. So individual genes were not the best way to assess antibiotic resistance in these communities. So here is just an example of a previous work from the group of Shah in 2014, when they observed, they did a similar work comparing aquaculture farm site and a control site with no salmon farms nearby. And they observed no statistically significant differences in antibiotic resistant levels in phenotypic assays using over 100 isolates from marine sediments and a salmon aquaculture site and a site with no aquacultural activities. And they found no significant differences in antibiotic resistant bacteria, both in number of bacteria and in the expression of antibiotic resistant genes. And here, when we use cultural...
Sorry yes. to interrupt you. You are 2 minutes before
okay.
the time.
Yeah, this is my last slide. Well, here, using the cultural bacterial communities, we observed a marked differences in antibiotic resistance between the Pacific Shore site or the control site and the aquaculture farm. In fact, the frequency of the resistant colonies was significantly higher in the aquaculture farm community, regardless the media use for the assays. This also correlates with an increased antibiotic-resistant genes abundance, but not when assessing individual genes, as I mentioned, but when they were grouped as a family. We hypothesized that growing bacteria in a community allows to obtain a higher diversity of bacteria, and which increases the probability to proliferate antibiotic-resistant bacteria as well. So this group explained this difference because they say that the antibiotics could leach from the aquaculture farm site to the control sites. So we used a longer distance that they use in this work, but we also use a different approach to evaluate antibiotic resistance, both in the isolates and the type of antibiotics supplemented. The sample size is also important because they used individual bacteria while we used consortiums and metagenomes, which allowed to observe a higher higher complexity of the community. And finally, I would like to add that the paradigm of the 1% culturable bacteria needs to be revisited, because more than consider unculturable organisms as a technical issue, we should consider the different techniques as complementary, acknowledging their biases, but using also their advantages. And I would like to thank my collaborators and everybody that participated in this work. Thank you.
Thank you very much, Javiera.